Hi everyone, I hope you are doing great. In continuation to our discussion about the secondary structures of proteins, we are going to extend our discussion today in the form of beta sheets, turns, and loops. Now, in our last class, we discussed about the alpha helices. Those are the most common secondary structures. So these beta sheets, turns and loops, they are not as common as helices, but they are as important as helices because all these secondary structures they make the foundation for the three-dimensional structure of the protein. So let's start our discussion. Now, beta conformations they are a second type of repetitive structures, okay, and they are more extended conformation than helix. So if you compare the structure of the helix with this particular sheet, you will find that the beta sheet has more extended conformation as compared to the helix. But like helix, they are defined by the backbone atoms and which are arranged according to their characteristic dihedral angles phi and psi. That is, we can use the Ramachandran plot. Okay. Several beta strands they align side by side and they are stabilized by the hydrogen bonds between the neighboring polypeptide chains, not within one. Now, this is really very important difference between the helix and the sheet. In case of helix, you see here that the hydrogen bond which is making here it is between the residues within the same helix, within the same peptide chain. But if you can see here in case of the sheet, the hydrogen bond is between the neighboring chains. Okay, so that is the very important difference between the helix and the beta sheet hydrogen bonds. Now, the tetrahedral arrangement around the carbon atoms and hydrogen bonding, it makes the sheet appear like pleated. Now, you must have seen some pleated sheets, and in our childhood also we make and we used to make such kind of sheets there by repeated folding. Okay, now how it is made you can see in this diagram or in this picture. You can see here this is a flat paper, but when you fold in this particular manner as shown and you release it, then it becomes pleated, and that's how the name comes the pleated sheets because of this appearance. So the beta pleated sheets they appear like this particular folded sheet, and that's why here they have the extended conformation. Okay, now in a beta sheet each main chain is called beta strand, and this beta strand is fully extended, and the side chains of adjacent amino acids they point in the opposite direction. So you can see here in this particular picture side chains they are pointing opposite directions when they are adjacent to each other. So this is adjacent to this particular residue, and you can see here they are opposite, these two are opposite, these two are opposite, and so on. Okay, now several beta strands they align side by side and then they stabilized by hydrogen bonds and they form beta sheet. You have seen here. And we have two forms of sheet. One is the anti-parallel beta sheet where the neighboring hydrogen bonded chains they run in opposite direction. So you can see here this is the anti-parallel beta sheet where these arrows they represent the beta chain and they are running opposite to one another. So you can see here if this is up, the neighboring chain is down, and then again the chain is up. So this is known as anti-parallel beta sheet. So you can see here how they are folded. Okay, now the parallel beta sheet, the neighboring hydrogen bond chains run in the same direction. So you can see here all these three chains they are running in the same direction, and therefore they are the parallel beta sheet. So you can see here how parallel beta sheet is formed, and a loop is required, a 180-degree loop is required to make this parallel sheet here, right? But here we don't need 180-degree loop. So that is also the major forming factor of this parallel and beta anti-parallel beta sheets. So if we compare the parallel beta sheet to anti-parallel beta sheet, then in case of parallel, the hydrogen bonding is distorted. So you can see here hydrogen bonding is going slightly up, down, slightly distorted. By here it is straight hydrogen bonding. You can see here hydrogen bonding is pretty straight in case of the anti-parallel. Okay. Then parallel beta sheets are comparatively less stable as compared to the anti-parallel beta sheets. Okay. The third difference is that parallel beta sheets, they appear more twisted, while the anti-parallel beta sheet, they appear relatively straighter. Why? Because particularly of this hydrogen bonding, which is straight in case of the anti-parallel beta sheet, while it is slightly distorted in case of the parallel beta sheet. Now, the repeat period is 6.5 angstrom in case of parallel beta sheet, while it is 7 angstrom in case of anti-parallel beta sheet. So this is something which has been determined experimentally. The phi angle in parallel beta sheet is minus 119 degrees and psi angle is plus 113 degrees, while the anti-parallel beta sheet, it is minus 139 degrees and plus 135 degrees. So you can see here the difference in the phi and the psi angles in this parallel and anti-parallel sheets. Okay. Now beta sheet, it consists of around 4 to 5 beta strands, right? And it can be either all anti-parallel, or it can be all parallel, or it can be mixed. Some are anti-parallel, some are parallel. And it can be relatively flat or some twisted. So there is a diverse structure in the beta sheets. They are very harder to engineer than alpha helices. Now when you engineer protein, okay, protein can be engineered. So protein engineering in itself is a big course. So when you engineer a protein, then these sheets are harder to engineer as compared to the alpha helices. Now why it happens? Because they are structurally more complex than alpha helices. We know this. They are prone to sticking together, forming clumps. So they can form clumps. And in many pathologies, for example, Alzheimer's and Parkinson's, amyloid fibers, they clump together, they stick together, and they cause the disease. So this clumping is very much seen in case of the beta sheets as compared to the alpha helices. Then the beta strands can align in many ways because they are strands. So not necessarily they can be parallel. Sometimes they not be parallel. Okay. So they can align in many ways when we are engineering beta sheets here. Then parallel versus anti-parallel arrangements differ. Okay. So these two arrangements, they differ. And we know very well how these arrangements are done using the turns. Okay. Now side chains alternate above and below the sheet, so that also makes slightly difficult to make these sheets to engineer than the alpha helices. And then the distant parts of the sequence, they may come together. So again, all these factors, if we combine, then our helices are easier to engineer as compared to the beta sheets. Now let's focus on the turns and loops, which are again very important secondary structures. So in compact folded proteins, some amino acid residues form turns or loops where the polypeptide chain reverses direction. So you can see here when they are reversing the direction, they form turns, and when they also join to some other secondary structure, they can form slightly loops or maybe turns. So basically they are the flexible linkers, okay, between the structures here. So the connecting elements linking successive runs of alpha helix or beta conformation, they are the turns and loops, right? So they are also very important structures. Now turns comprise an consecutive residue, therefore it is denoted I to I plus n. We don't know how many residues are there. It may n may be different number, different positive integer. It may be 2, 3, 4, 5, 8, 10, right? So it is denoted I to I plus n. So the distance between the C alphas of residue I and I plus n, it must be smaller than 7 angstrom. Remember, and in some cases, some literature suggests that it may be smaller than 7.5 angstrom. It's okay, 7 or 7.5, but majority data, it is in the support of 7 angstrom. So that means if we have this constraint, because we cannot go beyond 7 angstrom or 7.5 angstrom, so we have some structural features which will come up because of this. Now how? Let us see here. Turns can be gamma turn. Now gamma turn is when n is equal to 3. So this kind of turn is called as a gamma turn. Then if n is equal to 4, then it is called as a beta turn. If n is equal to 5, then it is called alpha turn, and then if n is equal to 6, then it is called pi turns. So we can see here that the turns can be gamma, beta, alpha and pi depending on what is the n, the number of residues here. Alright, and you have to remember that the distance between the C alphas of residue I and I plus n, it must be smaller than 7 angstrom. Okay, so the C alpha atom of residue I and the residue I plus 3, it must be close together through either equal or less than 7 angstrom when we are talking about the gamma turns, or sorry, when we talking about the beta turns, because it is I and I plus 3. So I plus 1, I plus 2 and I plus 3. So total n is equal to 4. So this is the beta turns. The protein backbone, it cannot remain extended. As I told you that we will see how this small distance can cause such features. So the protein backbone, it cannot remain extended. Okay, so it is forced into specific arrangement of bond angles and dihedral angles, causing the polypeptide chain to bend and reverse its direction. Why it is happening? Because we have a constraint of 7 angstroms here. Okay, so now beta turns, particularly we are talking about the beta turns. Out of these 4 turns which we have seen, the alpha, beta, gamma and pi, so the beta turn is the third most important secondary structure after the helices and beta strand. Remember, it corresponds to approximately 25 to 30% of all protein residues. So data experimentally again, the most common structural element that brings about chain reversal in proteins is the beta turn. Okay, the most common structural element that brings about the chain reversal in the protein. So that means the chain which is going up, it is going now down. So it is known as chain reversal. Now you must have heard about the beta turns, beta bands, tight turns, reverse turns or Vankert-Chalom turns and hairpin terms. So these are all different terms you must have heard. So beta bands or beta turns or reverse turns or hairpin turns, they often treated similarly. Okay, they often treated similarly. So you can see here, this is the hairpin turn which is reversing the peptide. Then this is again a slightly turn which is loop. L1, L2, L3, these are the loop. Usually we call them as the loops. Now remember that every beta turn is a reverse turn, but not every reverse turn is a beta turn. Okay, every beta turn is a reverse turn, but not every reverse turn is a beta turn. Then tight turn, okay, this particular tight turn, it is a broader term which describe any sharp directional change. So it can also be seen as a tight turn. This can also be seen as a tight turn. So wherever there is a sharp directional change, not necessarily 180 degree, then we call it as a tight turn. Okay. Then Venkachalam turn, it refers to classically geometrically defined beta turn, and it is of types like type 1, type 2, 1 prime and 2 prime classifications. So there are different kind of these Venkachalam turns. They are basically classical geometrically defined beta turns, because Venkachalam was the scientist who was behind the classification of these type of turns, and he worked extensively in 1960s and 70s. Okay. The stranded beta turns, they are classified by the backbone dihedral angles. So again, the dihedral angles phi and psi, they are used for the classification of the standard beta turns. And these represent the sites of chain reversals and important sites. Okay. Now they have non-periodic nature and the heterogeneity of their structures, because again, it is not that much, these terms are not repetitive, so they are non-periodic nature, and the heterogeneity of their structure, not necessarily they have one type, but they are different types. They are classified based on the different angles, and they have a heterogeneity of their structures because of the different residues which make these turns. All right. Now, in beta turn, the hydrogen bond, it occurs between the I and I plus 3 residue, and therefore it makes 180-degree turn. So beta turn is 180-degree turn. So Venkachalam proposed two major classes of the 4 residue beta turn. How many residues are there in beta turn? 4 residues. Remember, it is residue number 1 is I, then I plus 1, then I plus 2, then I plus 3. So total 4 residues in the beta turn. So type 1 is the most common beta turn. You can see here, this is the most common beta turn, and this purple color is the R group. Red is oxygen, blue is nitrogen, black is carbon, and white is hydrogen. And this is the hydrogen bonding, right? This dotted line is the hydrogen bonding. This is type 1. And type 2 is the second most common beta turn. So you can see here, this is type 2. Same labeling here, but the different orientation. So you can see here in this particular thing, check the difference here. There is a different orientation of the molecules. So that is type 2. Okay. Now, if we compare type 1 to type 2, then residue number 2, particularly this is 1, this is 2. We are talking about this. This is residue number 2. This is C alpha 3, third, and this is C alpha 4. Residue number 2, so we are talking about this particular residue. The phi is minus 60 degree and psi is minus 30, and it is often proline. Remember, it is often proline residue. And in type 2, this is again the residue number 2. We are talking this one. Phi is minus 60 degree and psi is 120 degree, and again it is often proline. So you can see here at the position number 2 in the beta turn, it is usually the proline. Okay. Now let us focus on residue number 3. This is the residue number 3, and here this is the residue number 3. Phi is 90 degree and psi is 0 degree, while here it is phi is 80 degrees and psi is 0 degree. So in both the cases, the psi angle is 0. So the type 1 has a smooth chain reversal. All right. Type 1 has very smooth chain reversal, and that's why it is the most common beta turn. But the residue number 3 in type 2, it is positive. All right. And you can see here, this is minus 90 degree and here it is positive 80 degree plus 80 degree. So residue number 3 is positive 5. So there is a steric hindrance. Okay, and because of this, usually glycine is at the residue number 3, which allows extended conformation. Therefore, type 2 is also called as glycine turn. Remember, type 2 beta turn is also called as glycine turn because it is present at the residue number 3 to compensate or to remove the steric hindrance. Okay, that's why this is known as the glycine turn. Now, what are loops? Let's talk about the loops after turns. So these loops, they are the irregular segments of a protein that connect regular secondary structure elements like alpha helices and beta sheets. So basically they are the connectors and they are irregular segments, remember. So you can see here, there is a loop which is connecting the alpha helix here. There is no repeating patterns of the backbone hydrogen bonding. So there is no repeat pattern here. It can vary greatly in length, okay? And they act as a flexible linker. So ultimately they are acting like a linker. And a loop is often exposed on the protein surface. Remember, often exposed on the protein surface, and therefore it can interact with water, it can bind other molecules, or it can participate in signaling. So loops are very important because they can do a number of functions for a particular protein. All turns can be considered short loop reasons, but not all loops are turns. Again, this is something important because turns are ultimately, you can consider that they are like loops. But then you have to remember that all loops are not turns. Okay. Now let's talk about the hairpin loops. They connect adjacent beta strands. So we call them as a hairpin turns also. And then we have another example is the omega loops. They are typically 6 to 16 residues long, quite big, and they resemble the Greek letter omega, and that's why the name is the omega loop. So you can see here this is 71 to 85 omega loop forming omega. This is 40 to 57 omega loop, okay, and here it is 20 to 30 omega loop. So this is the residues numbers, and you can see here they very much resemble omega, not actually like omega, but resemble omega, and therefore they are known as the omega loops, and they are often found near the active sites of the protein or enzymes, okay, because they can interact or with uh, other molecules, and they can also interact with water. So these loops are very important for the linking secondary structures, right? So thank you very much for watching, and this was the discussion about the secondary structures of proteins, particularly the beta sheets, turns, and loops. Now in the next class, we will move forward in our discussion, and we will talk about the tertiary structure of proteins. Or maybe before that, I can take 2-3 examples of the proteins like collagen or some other fibrous proteins to understand how these secondary structures contribute in making the proteins. Till then, you take care of yourself. Bye bye.